Welcome to STAT's virtual event, Biomarkers, the Foundation of Personalized Oncology, sponsored by Astellas. Thanks for joining us. I'm Angus Chen, cancer reporter here at STAT, and I'm sure you all know how this works by now, but before we get started, there's a few quick housekeeping items. So first, at the bottom of your screen, you should see several icons. Use the chat feature to chat amongst yourselves or to send a message directly to the STAT team. Please don't put your questions for us in the chat. If you do have a question, that you would like to ask us during the event, please use the Q&A feature to type your question, and we'll try to answer it live. In the Q&A window, you should also be able to see the questions that other attendees have asked and upvote the questions you would most like to see answered. We'll be taking questions throughout the chat, so please don't wait until the end to submit. This is part of what makes these events so engaging and fun. We want to hear from you, and thanks again for being here. Before we get started, I'd like to welcome Ahsan Arozula, Senior Vice President and Head of Development Therapeutic Area at Astellas, for a sponsored introduction of today's event. Oh, you're still muted, Ahsan.
Thank you to everyone for tuning into this virtual discussion today. I want to share my gratitude with Angus and the entire team at STAT for organizing this event. I'd also like to extend my appreciation to the expert panelists for sharing their insights with us today. My name is Ahsan Arozula, and I'm the Senior Vice President and Head of Development Therapeutic Areas at Astellas. On behalf of Astellas, we're honored to be here sponsoring this event and kick off today's thought-provoking discussion on biomarkers and how they can play a role in the cancer treatment landscape. Unfortunately, most of us have been touched by cancer at some point in our lives, whether through family, friends, co-workers, or within our professional lives in healthcare. At Astellas, we are focused on a variety of advanced and rare cancers and are dedicated to advancing our research towards potential treatment options. This includes gastric or gastroesophageal junction, also known as GEJ cancer, where a diagnosis of advanced cancer can be challenging and difficult for patients and their families. Gastric cancer is the 5th most common cancer globally, with more than 1 million cases diagnosed in 2020. And in the US, more than 26,000 people are expected to be diagnosed with gastric cancer in 2022 alone. This type of cancer is particularly challenging since it's often diagnosed once it's reached advanced stages. Initial signs that lead to diagnosis are often similar to common symptoms of minor stomach ailments such as nausea, heartburn, indigestion, stomach pain, poor appetite, or simply feeling full after a small meal. For patients diagnosed with gastric cancer in the metastatic stage, meaning that the cancer has spread from the tumor's origin in the stomach to other bodies or tissues, unfortunately, the 5-year relative survival is approximately only 6%. These devastating outcomes speak to the high unmet need for these patients and their families. And while progress has been made in recent years, treatment options for gastric cancer patients are still limited. That is why Astellas recognizes the important role precision medicine can play to advance cancer care. One avenue is through novel biomarkers, as they can be the key to unlocking more opportunities and new approaches to treating cancers. Different biomarkers are currently being studied within a variety of cancer types. Biomarkers are molecules that are found within the blood, fluids, or tissues of the body that indicate a normal or abnormal process or condition or disease. Broadly, there are multiple types of biomarkers, which can include established biomarkers that have been well studied in scientific clinical trials, as well as emerging biomarkers, which are currently being investigated in studies across the industry. Biomarkers have the potential to be important in several cancer types, including gastric, breast, lung, and colorectal cancers. Zooming in on the gastric and GEJ cancer landscape specifically, as novel biomarkers emerge, they may reveal more opportunities to advance care for patients with metastatic gastric or GEJ cancer. Expression levels of more established biomarkers like PD-L1, HER2, and MSI-high dMMR have been studied and detected in gastric cancers. For example, HER2 positivity has been reported in 22% of advanced metastatic gastric or GEJ cancers. Additionally, Claudin 18.2 and FGFR2b are emerging biomarkers in gastric cancer. And recent studies have shown that while approximately 70% of locally advanced or metastatic gastric or GEJ cancers express Claudin 18.2 at any level, approximately 36% of these cancers meet the qualification of being Claudin 18.2 positive or having a high expression level. Additionally, FGFR2b has been observed in 30% of patients with gastric cancer. In our research, Astellas is currently investigating treatments that target Claudin 18.2 and its potential applications in gastric and pancreatic cancers. As research continues in emerging biomarkers like Claudin 18.2, it has the potential to expand our view of the patient population, reveal more information about the gastric cancer landscape, and can help inform clinical decisions. We appreciate that no two patients are the same, and each tumor may have individual characteristics which can be identified through biomarker testing. It's important to increase access and availability, as initial diagnostic panels that include biomarker testing can help map the path forward for patients and lead to more informed clinical decisions by physicians. This is why it's important to continue to explore how biomarkers can play a role for cancer patients, and we are committed to research that can identify potential new opportunities through exploration of emerging biomarkers like Claudin 18.2. To learn more about the role of established and emerging biomarkers in gastric cancer, I encourage all of you to visit www.gastriccancerbiomarkers.com for more information. Like me, I hope everyone is looking forward to listening to an insightful discussion from the panel who can speak to the role of biomarkers in oncology. Thank you again to everyone tuning into today's event. And now back to Angus and Stat to lead the live Q&A panel.
Thank you, Austin, for that introduction. So here to join me for today's discussion, please welcome Dr. Ann Barker, Chief Strategy Officer at the Lawrence J. Ellison Institute for Transformative Medicine, Dr. Gabe Kwong, Associate Professor and Wallace H. Coulter Distinguished Faculty Fellow in the Department of Biomedical Engineering at Georgia Tech and Emory School of Medicine. And finally, Dr. Edith Perez, Chief Medical Officer at Bolt Biotherapeutics and Professor of Medicine at Mayo Clinic. Thank you all for being here. So I wanted to start today's discussion off by asking each of you kind of what makes you the most excited about biomarkers in cancer, and what's the great potential of next generation biomarkers to you? And we'll start with you, Anne.
Oh, that's a long answer. First of all, people who know me know that I'm particularly excited about biomarkers, and I've spent a fair amount of my career actually trying to sort out not just what's, why they're important, but also how do we make them better. And so I think my feelings right now are we've spent a lot of time getting to the point we are, and the point we're at is probably revolutionary in so many ways, because we're getting a reasonable handle on the digital information involved in cancer, and increasingly we're sort of closing in on some of the analog information. So and that they're both important, and we're starting to put that together in ways that allow us to look at biomarkers differently. And there's a very specific definition for biomarkers by FDA and NIH, as you all know. But I think we're at a point in time where we're going to redefine biomarkers, and they're going to be reflective of systems. They're going to be reflective of the complex system that is cancer. And doing that's going to require some rethinking of how we approach biomarkers. But I'm very excited about that. And I think what you're going to hear today is the future looks very bright. I mean, we focused, primarily on the genome for a while. Now, all of a sudden, we're looking all the way out beyond the RNA into, sort of what do we mean when we talk about epigenomics and its role. And it's very critical now in terms of talking about this for a very early detection of cancer. So I think it's, I just think it's the best time in the world to be in this space. And I'm very excited about what's possible.
Edith, what are your thoughts?
I tell you, first of all, I'm very happy to be part of this panel today. Seeing friends, meeting new people, and discussing this critically important topic, for science and patient care. For me, it started, actually, I can say a long time ago, and I always remember, having, I'm just going to tell a patient's story. A patient with very advanced metastatic breast cancer, at the very beginning of my career. And, we kind of ran out of agents to manage these patients. And then HER2 was identified. And then anti-HER2 therapy became available. And we were able to, utilize the new knowledge from one biomarker that had been discovered while she was still alive, and we were able to change her course of her life because we took advantage of the scientific knowledge that had translated into the development of a targeted agent. So for me, this has resonated, over the years. You just don't know when the next discovery is going to occur that would allow you to adjust your thinking on what therapy to recommend for a patient. What clinical trial might a patient be eligible to participate in? Because a new biomarker has been identified or potentially identified that may be predictive of benefit while minimizing the use of ineffective therapies. So to me, that's the center of biomarkers. It really, really aligns with people's quality and duration of life. And it ultimately, I think, saves a tremendous amount of money if we can identify the right biomarkers to make the therapeutic decisions that are critical for individuals. So I'll stop there.
Thank you. And Gabe, what do you think?
Yeah, well, first, I also want to say thank you very much for inviting me, and I'm thrilled to participate in this conversation. For me, I am an engineer, and I am actually most excited about the convergence of sciences as we move forward, right? So historically, you think about a lot of biomarker discovery, it's genomics, it's proteomics. These are tough areas, especially in the context of earlier cancer detection. And so what we're seeing in the past 10, maybe 15 years is we're attracting researchers from engineering fields like bioengineering or have a background in synthetic biology. And now we're bringing in new tools, right? Building genetic circuitry or new types of sensors that can pick up or report on cancers that previously may have been undetectable with a traditional naturally shed biomarker. And of course, I'm sure that we're going to touch a little bit on the concept of synthetic biomarkers as we move forward, but I'm most excited to see this future where clinicians and biologists and engineers are working together and all bringing in different pieces of the puzzle to really solve this really tough problem.
Well, I'm really excited about the discussion ahead for the next hour. But before we can talk about the future, I think it's important to establish where we are right now with biomarkers. And so this is a question for all of the panelists, but I'll prompt you with something that that Anne, you mentioned once, that biomarkers haven't been as successful as people think they've been. So what is the current state for biomarkers in cancer? And what are some of the limitations that we're running up against right now in our search and our innovation in this field?
So I just wrote a piece for this a couple of months ago for cancer research, and I was thinking as you were talking today, we have made progress. I mean, there's no question we've made progress. And I come back to something Edith said. I didn't remember when Herceptin was introduced. And today we have an example of a trial that we started in 2009, the I-SPY2 trial, which essentially is as much a biomarker trial as it is a drug trial. This is a Bayesian statistics-driven trial, essentially that's in the neoadjuvant setting. And essentially, the Bayesian part of it allows us to learn from every patient. And so what we did in setting this trial up was to make a deal, if you would, with FDA that we would settle to start the trial with the biomarkers that we knew were important. At that time, it was hormone receptor status and HER2. That was about it. And now we are able to, after it's 11 years, this trial's been ongoing. It's a platform trial. It can go on forever, actually. That's the beauty of these trials. And you see that now. And a lot of the work we've done during the pandemic has been in the adaptive space now. So the interesting thing that's happened, though, in this decade is that we are now able to subtype this disease. Not clearly yet, not absolutely yet, but enough so that we can predict who is going to respond and who's going to respond. And more than that, we have established through this trial, we established a surrogate endpoint, and that's critically important for biomarkers. If you're going to actually do clinical trials, you need surrogate endpoints. Surrogate endpoints predict how patients are going to actually respond to a drug. Insofar as you could do that, that's hard. There aren't many surrogate endpoints actually in oncology. This one is pathologic complete response. But we can now subtype that disease, this is neoadjuvant breast cancer, such that we can make pretty good predictions about who's going to respond and who isn't. And that's, as Edith said, that's worth its weight in platinum, because you're going to get the right drug to the right person. That's the whole mantra of precision oncology. And the same thing's true in colon cancer. You know, knowing that what happens with people who, you know, who have KRAS mutations, for example, are not going to do well. We have gene panels now that can tell you, don't even bother with this drug. You're going to have to go to another drug. So I think we've made real progress. I think, especially in genomics, I think I had a little bit to do with that with the Cancer Genome Atlas. We did set people up to discover, you know, what genes were important, and I'm happy about that. We're still struggling in the protein space, and I think we will continue to do so. And, you know, I always ask the question, are the protein biomarkers actually there? Are we going to be successful in this space? The technology gets better and better. So I think we've made an enormous amount of progress. I think current progress, though, is capitalizing on big data. And big data is really important for biomarkers. And it's not just any data, it's high quality data. And that's what I think our clinical trials, HER2 especially, has been shown over these years to be so heterogeneous across, not just across breast cancer, but across all cancers now. You can find HER2. And that's another thing that these studies have taught us, that these very important biomarkers are part of pathways that may very well be common to all cancers. So I'd say we made progress. I think we got a long way to go. And we may just jump over a few steps, as Gabe said. We may go to another way to look at biomarkers altogether, and I'd like to talk about that later.
I think, you know, I look forward to hearing from Gabe about synthetic biomarkers pretty soon, you know, but before, so, you know, in terms of the reality of where we are, it's a very complex field. Again, there are a few biomarkers, but increasing numbers that have been approved by the FDA to be associated with benefit to a particular treatment. And this is something that has grown over the years, so it's very gratifying to see that evolution. At the same time, there is no biomarker that I know of, maybe, you know, from Gable or Anna, any of you, that really predicts 100% that the patient will benefit. And that just is a reflection of the complexity and heterogeneity of cancer. Not only from the genomic, but the proteomic, but epigenomic, epigenomic or epigenetic changes that occur. So although we are getting better, you know, as you alluded to, there's still more work to be done. And another aspect in terms of the reality of what we are, that sometimes we think we understand things based on the preclinical models, but gosh, there's a big difference between mice and humans. In terms of the predictability of biomarkers. So as we do clinical trials, I think it's important to be humble and explore whether a biomarker that we thought would be predictive on preclinical models is really going to be predicted in the clinic, instead of just, you know, feeling, okay, it's going to be 100% because it worked in mice. So, and the other aspect of this, and again, I'm opening complete doors for both of you, is the issue that, you know, there's so many tests available out there that is actually becoming very challenging for clinicians to figure out what test to order for the patients, to make the decisions. And not only it's the fact that there's some FDA approved tests, but many of them are, you know, modified by the different institutions, which creates a little bit of a challenge. But the competition is pretty wild out there related to the availability of biomarkers. So I'll stop here and open it up.
Yeah, I mean, I couldn't agree more with my colleagues about the tumor heterogeneity problem, you know, that the biomarkers frequently aren't there. I think what would be super helpful moving forward, and a lot of this is actually starting to be done, is to put numbers to these, right? So like, what's the half-life of cell-free DNA? If you look at a protein marker that's being shed by a cell, like how much can a tumor cell actually make that protein, right? And you can put together quantitative numbers and put together models. And of course, there are limitations of how predictive a model could be. But I think we just need a deeper understanding of not just the complexity of the biology, but also like, what are the transport challenges, right? If you were a biomarker that just got shed, you know, magic school bus style, you sit on the biomarker and you say, what am I seeing, right? I need to escape the tumor bed, then I need to go into the bloodstream, and I need to circulate for long periods of time before I'm picked up. So these are all, and again, just to reemphasize that I think this is where engineers who are good with math, good with multi-compartment modeling can help us develop a deeper understanding of what are the true bottlenecks in each of these major steps so that we could figure out creative solutions to get around these problems.
Yeah. Well, I'm going to take a quick break and ask a question from the audience that came in. This is from Kyle Nixon, and he wants to know how meaningful is understanding the spatial context of biomarkers for drug discovery, development, or clinical trials. And referring to spatial biology, there's a couple of companies here: NanoString, Akoya Biosciences, 10x Genomics. I don't know if you're familiar with these companies, but I'd love to hear the panel's opinion on this question.
Well, I'm extraordinarily enthusiastic about this. The spatial and a lot of other features actually that we don't pay a lot of attention to. Imagers pay a little more attention, than most of our molecular friends. But I have those of you that know me know I also am very enthusiastic about convergent science, and I set up a program at NCI called the Physical Science and Oncology Centers, where we brought together physicists, mathematicians, engineers, and cancer biologists, and we learned an enormous amount about the importance of measuring these spatial considerations in cancer. And if you think about it, how are we still diagnosing cancer? We're looking at it under the microscope even today, but what's happening? Digital pathology is coming into its own, and you can see it as we speak, almost expanding to a point where I think in the future, it's going to be very much about the spatial aspects, because let's face it, the reason that we look so carefully at these cells, it's the shape of the cells that matter. It's the irregularities in these cells that really make them cancer cells. So the best way to look at that is through an engineering and potentially even mathematical perspective. I saw Larry Norton on this call or on this Zoom, and he thinks a lot about this and has thought a lot about it for years. It really does make a difference. So I'm hoping that the future is going to have spatial aspects.
That's great. In terms of this, spatial, analysis, I think it's really fascinating. But one of the calls I would have, for the different companies, because we've been, playing around, if I may use those words, with one of those technologies, with the last few years in the laboratory at Mayo, is that the companies that develop these tests, I think, should pay increased attention to the logistics of getting the test done the correct way. Such as how do you assign, the regions of interest that you will utilize to do the genomic or the transcriptomic evaluation of the different proteins that may be present in the stroma or within the tumor or analogous to blood vessels. So I think it's like everything in science. Methodology is important so that interpretation can yield the outcome that we're looking for. It's like, real world data, it's going to be a great tool to utilize, but the data that are entered need to be accurate. And that this applies to biomarkers.
Yeah, and I'll just add that I think the spatial component also reaches to therapies because, there are many classes of therapies where they require conditional activation, right, to reduce their off-target toxicity. And so if the source of the signal is clustered around a blood vessel or is heterogeneously distributed throughout a tumor, this will have direct implications on how you design a therapy. So yeah, I'm all for, understanding more spatially how biomarkers are distributed, and we could potentially use that information in new ways.
Thank you. So I have a question that I'm going to ask Edith. We talked a little bit about sort of the need for innovation in searching for new biomarkers. And I wanted to ask you what you see as sort of the most promising ways that sort of in the search for new biomarkers that seems to be yielding a lot of progress right now, or things that you think will yield a lot of progress in the future.
Collaboration.
Yeah.
That's, because none of us can solve this problem alone. None of us has enough biospecimens. to answer all of the questions. So it's something that Anna brought up. We need good data that are annotated. And then people need to be willing to share those data so that they can be aggregated like persons like Gabe, and to put the puzzle together. But this is something that is discussed a lot, the importance of collaboration, the importance of data sharing. And this is something that we can continue to stress, because for biomarkers, it's going to be as critical as anything else.
Yeah. And Anne, I wanted to ask you something that you'd mentioned a little bit earlier. In the beginning of this conversation, you said that we need to rethink our approach towards biomarkers, and we need to redefine biomarkers. Could you tell us a little bit more about that? Like, how exactly do you see that rethinking happening, or what is this new approach that you see?
Well, so I consider myself a complex adaptive system scientist. So I think a lot about system science and how things come together in three-dimensional space and time. And that's what humans are, by the way. So, we are, when we talk about the phenotype, we're talking about the whole phenotype, not just the genome, not just the digital information, but everything else. And that's why, we finally have paid attention to real world data from patients. It's really meaningful because it's coming from the entire interaction of a series of systems. So I think what will happen in the future, and here it gets a little wonky, but I think this is where we're going to go. It's taken a long time to get to a point where we can even say this, but we're creating a lot of data. And so that's empowering us to build some pretty good algorithms. And that's taking us, I think, a couple of steps down the road. What we're not doing, though, is welcoming in physicists and mathematicians to apply theory to our data. So the whole world of cancer biology has no theory, save evolution. We don't even think about evolution that much, actually, in cancer. We should think a lot more about it, and we will. But I think it's going to move to applying theory to the data that we have and be able to turn that into real information. Data is not information, it's entropy until it has context. So now that's wonky, but that's physics. And data is a real thing, and information is a real thing. And we think of it as some elusive thing that's in the ether, but it isn't. It's a real thing. So I think the future is going to be looking at finding those critical biomarkers that represent the kind of convergence that Gabe was talking about. And it may very well be that they're more coarse-grained than we think they should be, because we've been studying the parts of cancer for a very long time. And we're sorted out. There's a great paper on how a biologist fixes a radio. We're doing what a biologist would do. We've got a bunch of parts lying around on the floor, but we're trying to put them together now. And I think you need theory to do that. And so I think the future is going to belong to how do we put this together in a way to allow us to understand the functioning of the system, and how does it relate to what's expressed in the phenotype? That's hard. That's really hard science. But without, coming back to Dave's point, without physics and math and engineering, we're not going to get there. So I think NCI is making some efforts in this area, and other organizations, but it's a reach. I think it's going to be hard, but I think that's where we have to go.
Yeah. I mean, well, so Gabe, you've really building a career on this type of collaboration that sort of looks at innovative ways to bring together different fields in biology and medicine and engineering. Synthetic biomarkers, I think, is one really cool example of that. So for the uninitiated listening in, maybe you could tell them first, what is a synthetic biomarker, and how do these sort of start to tackle some of the challenges that we've talked about on this call so far?
Yeah, of course. So the synthetic biomarker framework is just a recognition that there are limitations with a traditional tumor shed biomarker, right? So especially in the context of earlier cancer detection, you can do the math. There aren't that many molecules, for example, of DNA that a tumor cell, that a tumor bed can shed, right, for early detection applications. So a number of years ago, myself and other labs across the country started thinking, as engineers do, this is a problem. So how do we create a solution that maybe it's not as traditional as what we've seen before in papers prior? And so the thought of a synthetic biomarker is this thought experiment. Well, since a tumor-shaped biomarker has these limitations, can we make a synthetic biomarker that could overcome some of these limitations? So for example, if a protein circulates for short periods of time, can we create a synthetic biomarker that extends half-life? Okay, so that's like one simple example. If, as we know, proteomics is really tough because blood is so complex of all the peptides and albumin, IgG in it, can we have the signal be concentrated into the urine, which is a much simpler matrix? And so once you think this way, the framework is essentially, okay, we can design sensors that we send throughout the body, and that these sensors would query sites of early disease. It could be organ, it could be tissues. And then in response to early disease signals, tumor cells, it would then produce and drive the amplification of production of the synthetic biomarker. So at its high level, that's the vision.
Yeah. Well, do you have any examples of like a synthetic biomarker currently in development that sort of really excites you and something that you could potentially see moving forward? Yeah.
Yeah. So what we've been doing in my lab for the past 5 to 10 years, which branches off the work done by Sangeeta Bhatia at MIT, is the designing of chemical probes. So these are akin to contrast agents that you develop at the bench, but they're designed to be administered, and that these probes are cleaved by tumor proteases. And when you move into the synthetic biomarker framework, what's interesting, now we can start to think of strategies to amplify the detection signal, the on-target signal. You think about strategies to improve specificity. So for example, if you were to take one probe and you inject it, well, it's going to light up, right? In many different tissues, like tumor proteases aren't that specific. But if you start looking at combinations of probes, right, you can start to build up a signature. We're not quite there to the big data, conundrum that Anne described. We're not generating that voluminous amount of data, but you can certainly scale up to maybe 10, even 20 biomarkers at a time, at least in animal studies, and create this synthetic signature.
Yeah.
Can I ask Gabe a question just quickly? So
go ahead, please.
So we have now I'm curious because I've read his work and I'm just wondering. So one of our issues, obviously, is when we look at the genome, is the long tail, and we know it's important. We know there are a lot of genes out there that people call noise, but we know they're not noise. Mm-hmm. So the prevalence, so if you look at the prevalence of a biomarker, that's the limitation, as you point out. So is there a way, though, that you can start to use your approach in terms of synthesis of probes to begin to prioritize that list? I mean, it's if you have something and the prevalence is high enough for detection, you're good. But most of our problem is in those things we can't detect. The prevalence is too low.
Yeah, I think that's a tough, that's an analytical challenge. We like to focus on not just the abundance of a species, but the activity. And there's a huge difference between the abundance versus an activity, especially in the context of proteases, right? There are circulating inhibitors. So just because you can pick it up via ELISA or mass spec, it may not necessarily mean that there's some active inflammation, for example, going on at a particular site. So I think, it would require a combination of us to measure certain things like abundance, and if we can't detect it, perhaps we should switch to activity, right? Because again, activity, when it's enzymatic, it also tends to amplify your signals over time.
That's a good point. Thank you, Gabe. Thank you, Angus.
Oh yeah, of course. Well, I wanted to, before we move on to the next topic, I mean, Edith, do you have any thoughts on sort of this idea or any other points to bring up?
Hey, my enthusiasm is increasing here. It's good to be innovative and bring different disciplines into looking at this challenge and how to solve it.
I'm going to ask another audience question. This one comes from Pei Zhang. What do you think about serum/plasma biomarkers for cancer risk prediction and early cancer detection compared to the tumor markers? If anyone from the panel has an opinion on this question.
Could you clarify the question a little bit? Is he asking about the serum for pathologic diagnosis? What I don't understand what he's asking.
Probably the Grail. They're asking about the Grail test, or which
is he asking Grail? Yeah, that's what I think he's
My understanding was about maybe serum or plasma biomarkers, maybe like the GRAIL test that I think looks at the epigenome, right? Or epigenetic markers,
Right.
sort of things that you might see elsewhere in the body versus things you might find that's either directly shed by tumor cells or found on the tumor cells themselves, the actual tumor cells.
Well, I'm not sure it makes a lot of difference, but I will say I think these tests are going to be. We were talking about this before we started today, and I actually think we're in the infancy of this whole movement, and it's kind of going to be a movement because this moves the needle back toward either very early detection means you may be able to downstage a cancer by finding a stage 2. You may be able to. I don't think will do well at finding very early cancer. I don't think we will. And I think that's going to cause problems. I think they'll easily find stage 4 cancer. It's not a bad thing. If you can find it earlier, you can downstage it. I think it's very promising, and it's starting out in the methylation space, but it's starting to be picked up now by messenger RNA experts. And I see today, even in STAT, there are lots of people thinking about, circular RNA. So I think there's all kinds of possibilities here, and I think we better pay attention to it. Because it does create a series of problems, as you know, Angus. If someone thinks they're going to get cancer, but we have no way to give them anything or to reassure them or know what to do, that's going to create a whole bunch of different issues. And so I think it'd be very difficult to sort this out. I mean, but it also opens the door to tumor evolution now, because I think people can start to study these clones in ways to be more predictive. So I think there's no question this door is open, and we've taken one step on this road, but I think there's a lot to learn yet. But I think it's something we should do.
I would love to follow on this, because to me, I see this as a continuum of the whole screening strategies that have been used for detection of cancer for many, many years. Started with mammograms, and people, then thought about chest X-ray to find lung cancer early, and then CT was identified to be an appropriate screening tool to identify early cancers, and this has moved into in lung cancer bronchoscopies and looking at cells. And in the GI world, doing endoscopies to look at polyps, to try to identify not only early cancer, but pre-malignant conditions that we can alter. So as I look at these blood markers, I completely agree with what Anne just mentioned. It's really promising. There are some issues, though, that we should think about. Number one, I think there's about 140,000 prospective study, either initiated or soon to be initiated in Great Britain, to look at this particular one that we mentioned a few minutes ago. Because for screening and detection of early cancers, we have to think of not only the sensitivity and specificity of the test. Second, how often are you going to do the test in a way that if you find an abnormality, you will be able to alter the natural history of the disease? That's also very important. It's more than just doing it one. When should you do it? And should you repeat it at some particular interval? And then the other aspect that comes in screening that is going to be very critical is, again, the sensitivity and specificity. Because what if somebody has, some shed tumor cells that mean nothing, quote unquote, in terms of the development of an invasive cancer? So I'm very happy that there's so much effort being placed in this area to move beyond radiographic tools that only allow us to find tumors when they're more than 5 millimeters in size. And again, I'm very supportive of the prospective studies that are being done in this area. But there's some issues that we need to think about before implementing these early technologies for all patients as a way to find cancers.
Yeah. Gabe?
Yeah, and I think it's also interesting that we could combine some of the radiographic approaches where we're not really doing anything to help to force the production of a release of a biomarker. But there are actually a number of labs looking at how, if you take a blood measurement, typically it's what we call quasi-steady state, right? It's just the biomarkers might be there, but it's very low levels. It's not really changing much over time because the tumors aren't growing very fast. But there are labs that are looking at using imaging like focused ultrasound to disrupt what could be perceived as a tumor lesion, right? And then looking at the contents as a way to genotype it or a way to enhance transiently the amount of biomarkers that shed into the blood for a more definitive diagnosis. So I think there are exciting approaches that's, again, it's leveraging a little bit of technologies which we typically may not think is associated with a blood test.
Yeah. So there's another audience question that I think this discussion segues pretty well into. This might be two separate but related questions. This comes from Sinwan Basharat. What is the risk of overdiagnosis from biomarker testing? And what are the healthcare challenges of introducing tests that aren't cost effective and driving up healthcare costs? If anyone here wants to take this one on.
I think it's a really interesting practical consideration. Anytime we have a new technology, the same thing's true with therapeutics. You saw that the world is trying to do something about the cost of, of drugs now. And this will be no different. I think it's a good point. It would have, I think if the trial that Edith mentioned, that 150-person trial, which just finished enrolled, it was enrolled, I think, 2 weeks ago, completed. The data will probably be out within, I think, next year. I think these tests are going to be on the market. But once again, I think, Angus, we have to be very careful that we're not creating technologies for the rich, that only those that can afford these tests will be able to have them. And so, I think it's a very major consideration as this field moves forward, that we don't end up with another situation where we have a technology that could be of high value, but the prices are going to make it available to a fairly small number of people who can afford it.
Well, and it's bringing me, to think about the issue, not in the detection of cancer realm, but in the prognostic and predictability of biomarkers out there for patients already diagnosed with cancer, and the limited number of pathologists in other countries compared to the number of pathologists per 100,000 in the United States. And also, the impact of the College of American Pathologists in terms of, guidelines that help with the pre-analytical variables that are so critical to accurately measure or understand what the apparent results of tests are. And I'm so happy that you mentioned digital pathology, and I want to come back to this because this is going to be one of the ways to ameliorate the disparities that occur at the global level related to particularly the use of immunohistochemistry. And the issue of the cost of genomic tests, that's a completely a different situation where these tests are costing $4,000 or $5,000, which limit their use in many parts of the world outside of even within the United States, but particularly in other countries.
Yeah, and I'd just like to add, in the context of earlier cancer detection, it's a bit of a philosophical conundrum, right? If you do truly have an early detection signal, right, what are you going to do with it? Do you wait until the tumors grow big enough so you can validate by a biopsy or imaging, or do you treat and risk morbidity to the patient and stress to the patient, all that? So yeah, this is a real challenge for us, and I think, I'm not sure we know what the solution is just yet, but this is something we for sure as a community need to hash out moving forward.
I wanted to ask one of my own questions. We talked, of course, about synthetic biomarkers, but I was just really curious if the panel here thinks or sees other ways to combine synthetic biology with natural biomarkers to sort of achieve better precision medicine. And Gabe, maybe you could start.
Yeah, I'm actually quite excited. The idea of using living cells as sensors, right? So the synthetic biomarker field bore out basically from a biochemistry perspective, right? From development of imaging probes to now thinking about, okay, a biomarker that's produced from a biofluid. But we are in the advent of cell therapies, cell and gene therapies. And yes, the cost of cell therapies now are quite high, but like in any other industry, that price would drop. Right. So think about automobiles or sequencing. And so there could be a very unique moment in the very near future where potentially, even when you're healthy, we could have some subset of your cells engineered in such a way that they graft and they remain sentinels with you, right? And could support evidence of future disease. So I think that to me, it's a bit of a blue sky idea, but I think there's certainly excitement from my end to see this reality in the near future.
Anne and Edith, any thoughts?
Well, one of my dreams was a long time ago, way back in the early 2000s, we set up a nanotechnology initiative at NCI. And the idea was that, over time, we developed constructs, nano constructs, that actually could interrogate cells, could actually work within cells. And so, I mean, taking a little bit from Gabe's theme, what we thought about then, and I think what people are coming back to is, if you build these nanoprobes that are able to detect, can detect, let's say, an antigen from cancer, then what you'd like to have is the best biomarker on there. You'd like to have biomarkers that are probably synthetic. And then if you had a payload, essentially, to deliver, then the idea is that you can deliver it exactly to if it's a surface receptor or, wherever you're delivering a prodrug or whatever, then you can deliver it specifically. And I think that might be really attractive as we think about immunotherapy, where I think we're kind of awash in terms of not really having, biomarkers, even for picking patients who are going to respond to the checkpoint inhibitors. So, I think applications of nanotechnology could really use a synthetic approach, because the reporters we have and the platforms we have are too clunky. And I really think, Gabe, this is a place that could, the kind of stuff that Jim Heath used to think about, and others that I think a lot of people at MIT think about even now. So I think this kind of stuff is going to be the future. The industry is really starting to look carefully at this.
Edith?
Yeah, it reminds me, it's just complicated. Cancer, we're going to have to treat cancer, and it's clearly the field, with multiple tools. It just cannot be just one. And to me, that makes the situation with biomarkers a little more complicated, because it's not only one biomarker that is going to be predictive of everything that we need to know. It's just a multitude of biomarkers that we need to continue to investigate and interrogate. Mm-hmm. And there's something we haven't talked about. Perhaps I'll take this chance. And it is that we have to remember that we're asking volunteers, patients who participate in clinical trials, to give us specimens for both, the blood as well as tissue. So we have to think about the proper consent for the individuals, so that they understand, what they're agreeing to. Number two, for us to think to be as broad as possible in our consenting, because the field is evolving so much. And I think one of the problems that we've had is that in the past, the consents were so narrow that even though there are a lot of biospecimens stored in some freezers, they cannot be used because the patients were not consented. Go ahead, Anne, you're ready to say something about this.
Well, you will recall that when we started the Cancer Data Atlas, my major embarrassment is 30% of the samples in our country qualified for that project, because, as you pointed out earlier, we set up very, very rigid criteria, so we had quality data coming out the other side. And your point is so well taken, because you want truth in biology. You don't want to measure what anesthesia the patient might have had. So truth in biology is everything, and unless you get great samples, you don't have truth in biology. And that's one of our problems with biomarkers, is people use convenient samples, and that's a very bad idea.
Yeah. And the other aspect, if I may, on this is that in the area of cancer, obviously it affects everyone's lives, families, friends, in addition to the patient. And I think it can happen to anybody. And I think that if we do biomarkers in studies, I would like us to be able to share the information with the patients a little bit more. Even though there's still so much that we need to learn, but I would like patients to be more partners with us in this quest, instead of us asking, just give me your biospecimen, I'll do something with it. And then just, very, very little gets back to the patients. So I would like us to think about this a little more, send the information back to the treating physician to have the proper communication with the patient, because obviously in trials, we don't know the names of the patients. They're anonymous, they're just a number. But let's pay back here if we do some work and give back to the investigator who enrolled the patient in a clinical trial, because that information may help inform future approaches to managing that patient.
Yeah. And Gabe, do you have any thoughts on sort of this last bit of the discussion, and maybe how to involve patients or be more collaborative with sort of other groups?
Yeah, I think that's a good point. But maybe I'll just add a slightly different point, which is that in the synthetic biomarker arena, what is really needed is help as to what the first clinical entry point should be, right? So we have expert panelists here who run these amazing clinical trials and big data, and a synthetic biomarker approach is different because we have to assess safety, like especially in the context of multiple injections, and you have to think carefully what is the clinical problem that we want to first show that this idea works in humans. Screening is likely not going to be it. That's a really tough problem, right? It really requires a very high positive predictive value. But maybe it's drug monitoring, right? Maybe it's recurrence monitoring, and these are areas where seasoned vets, I think, can really shed their experience and help to nurture this nascent field.
One additional point, though, Gabe, would be, and it comes back to Ida's point, is patients deserve to know if they're in a clinical trial, what the outcome was and what the biomarker was. And one thing I would say that will be somewhat controversial, doesn't bother me. The issue with if a clinical trial doesn't work, we never know what the biomarker was. And I would really encourage the pharma industry to release that data, so that we get the biomarkers from that didn't work as well as the ones that did work. It would save enormous amounts of money and time and patient lives. And we've never been able to cross that bridge.
Yeah.
That'll be that's your assignment, Angus. Let's get that done.
All right. Well, I think we have time for maybe just one last question to wrap things up. And I wanted to ask the panel if there's something that this field needs to do to advance biomarker science or make this technology feasible, like in the clinic. Something that we haven't talked about already, maybe something to do with regulation or the industry environments or anything else, really.
So I'll start. There are two things that really I think could make a big difference. One is know what FDA is doing with biomarkers today. It's really quite different than it was even 5 or 6 years ago. Due to 21st century cures legislation, you can actually qualify biomarkers for drug discovery now and development, and there are other approaches to get your biomarker approved as a PMA, for example, so you can use it broadly as in screening, as Edith was talking about. And there are all kinds of ways to do that as well now with accelerated approvals and breakthrough kind of devices. So those doors are all open. The big problem with biomarkers, and I started a nonprofit to study this problem, why so many fail. And I just will tell you the quick list here is that they don't start with a relevant clinical question. They may start with a question that doesn't mean anything, but may mean something to them, but doesn't mean anything in the clinic. They don't collect the right samples. They don't have a robust design. They don't collect the right data, certainly not the right metadata, and they don't have the right analytics. And so that's why the literature is just fraught with, results that can't be reproduced. And we've got to change that. The biomarker scientific establishment has to become better at what they do, and they have to pay attention to quality.
Yeah, I completely agree. And I'd like to add that, I'm in an academic center, and so I see a lot of very young, smart and talented individuals come in and out of universities. And oftentimes, at least from my perspective, it's actually quite hard to get them excited about developing a new diagnostic or a new biomarker. And so I'm not quite sure whether there's a solution immediate in front of us, but given the choice between developing a therapy or a diagnostic, I would say more than the majority of my own students tend to gravitate towards developing a therapy. So I think that is something that we should definitely need to change, because this is equally as important, if not as important, right? We want to move from a situation where we're treating patients to preventing disease. And that requires a host new set of diagnostics to be developed. And so I think that's one challenge we need to address.
Absolutely. You know, I would, you know, share that, you know, right to, you know, some of Anne's comments at the very beginning, you know, things are getting better and they will continue to be better. And that's because some real thought is being placed into this, some different thought with open-mindedness, because we realize the importance of having best biomarkers, both for detection, prognostic understanding, as well as predictability of benefit. So let's continue to be positive. You know, I think the glass it's more than half full. Let's just keep going.
Well, I think with that last thought, we're just about out of time. Thank you, everyone. Thank you again to Dr. Barker, Dr. Kwong, and Dr. Perez for joining me today in this incredible discussion, and for answering the great questions from our audience. I'm so sorry we couldn't get to all of them. Thanks to our sponsor, Astellas, and to all of you out there for taking the time to join us. STAT's next virtual event will be part of our 2022 Status List Spotlight Series, which will be taking place on September 20th, when my colleague Usha Lee McFarling will be sitting down with Dr. Kirsten Bibbins Domingo, the incoming editor-in-chief at JAMA. More information can be found on our website. Thank you all again. Stay safe and be well.